My name is Palash Sashittal. I am from University of Illinois at Urbana-Champaign. I'm being advised by Professor Mohammed Al-Kabir, and I'll be talking about sampling and summarizing transmission trees with multi-strain infections. The COVID-19 pandemic has certainly brought to light the importance of computational methods that can facilitate real-time outbreak management and public health policies. One of the pivotal steps of these tasks is accurate inference of the transmission history of the outbreak. Traditional approaches of doing this involve fieldwork, interviews, and manual contact tracing, all of which are labor intensive and costly. But with decreasing cost of genomic sequencing, molecular epidemiology is becoming an indispensable tool in the analysis of disease outbreaks. Now, during an outbreak, the pathogen is transmitted from one host to another, all the while undergoing evolution, natural selection, and accumulating mutations. As a result, the evolutionary history of the entire outbreak can be summarized with one single phylogenetic tree, and the transmission history of the outbreak can be captured by a transmission graph, which connects hosts that infected one another. As an example, here I have shown the estimated phylogeny and transmission graph for the COVID-19 pandemic. So, although there's been a lot of development in this field, there are still several challenges that hinder the accurate inference of the transmission history. One of the biggest challenges is incomplete lineage sorting, where pathogen evolutionary history does not necessarily match the transmission history of the outbreak. This happens due to within-host diversity, which is the co-occurrence of multiple strains of the pathogen in an infected host. This is especially the case for pathogens with high mutation rates and diseases with long incubation periods. Further complications in the inference problems arise due to multi-strain infections through what is known as a weak transmission bottleneck. As an example here, host H1 infects host H2 via two strains of the pathogen. In the results section, we'll show that accounting for weak transmission bottleneck and within host diversity is essential for accurate inference of the transmission history. It's also important to discuss this because within host diversity has been observed in bulk sequencing samples collected from COVID-19 patients. So let me talk about the different patterns of transmissions that are observed during an outbreak. We have two axes here. On the horizontal axis, we have number of infectors for each host. So under unique infector, each host can be infected by only one person, whereas under superinfection, a host can be infected by multiple hosts. On the vertical axis, we have number of strains that are being transmitted during an infection. So under strong transmission bottleneck, a single strain is transmitted during each infection, whereas under weak transmission bottleneck, multiple strains can be transmitted. In our previous work, we proposed a method that looked at all these different patterns of transmissions and found the most parsimonious explanation for the transmission history. However, for some diseases, the unique infector hypothesis is known to hold. And so for these diseases in this work, we focus our attention on only the unique infector cases where the transmission graph is necessarily a tree. Previous works that have looked at this problem have focused on the strong transmission bottleneck, and so our contribution is to account for multi-strain infections as well. So let me introduce the problem statement. We call it the direct transmission inference problem. We start with the time phylogeny, which is a rooted tree whose vertices are labeled by timestamps. The leaves of this phylogeny are the pathogen samples that were collected from infected hosts. For each host, we have an entrance time and a removal time, and the host participates in the outbreak only during this time interval. So, for instance, the green host can only infect anyone or be infected by anyone in the time interval depicted by this green bar. The leaves of the phylogeny can be labeled by the host from which they were collected. So in this case, we have two samples collected from the red host and two samples collected from the blue host. No sample was collected from the green host, so green is an unsampled host. This is actually very common in ongoing outbreaks where you cannot have sampled all the hosts that have participated in the outbreak. Further, we have a contact map, which is a directed graph with the vertex set given by the set of hosts, and this graph shows putative transmission pairs. So the transmission graph that we would infer is necessarily a subgraph of this contact map. Now our goal for a given input is to color the internal vertices of this phylogeny and construct a resulting transmission tree, which is a spanning tree of the contact map. Each edge of the transmission tree is weighted by the number of strains that were transmitted from the donor host to the recipient host. So in this example, the green host infects through 2 different strains of pathogens to the red host, and so this edge is weighted with 2. Now it turns out that for a given phylogeny and epidemiological data, it might happen that you have multiple different solutions or multiple vertex labelings that result in a transmission tree, that is spanning tree of the contact map. And so natural question arises, given a problem instance, does there exist an internal vertex labeling that results in a transmission tree? And if so, how many such vertex labelings exist? So we look at the complexity of this problem, and we find that the decision problem is NP complete, and the corresponding counting problem is sharp P complete. We show this by reduction from the 1 in 3 sat problem. In this slide, I have shown, given a CNF formula, how we construct the time phylogeny and the contact map for the reduction proof. So now that we know that multiple solutions are possible, we use the following approach. Given an input instance of phylogeny and contact map, we uniformly sample from the solution space, and then we use a consensus-based approach to get a consensus transmission tree for the outbreak. So we need 2 sets of methods, one that can uniformly sample the solution space of a problem instance, and another that can summarize that sampled solution. So for the uniform sampling of the solution space, we use the SAT oracle. Essentially, we convert our problem into a SAT problem, which is kind of opposite of what we did in the reduction proof. So to do this, we introduce 2 kind of variables. One is an X variable, which encodes the host labelings of the internal vertices. So xi, s would be 1 if vertex vi is labeled by host s, and it would be 0 otherwise. Similarly, we also introduce c, which encodes for the transmission tree. It's another Boolean variable, which would be 1 if, so c s, t would be 1 if host s infects host t, and it would be 0 otherwise. So once we convert our problem instance into a CNF formula, we can use tools like Unigen and ApproxMC for almost uniformly sampling and approximately counting the solution space. So once we have sampled some transmission trees from the solution space, our next task is to construct a consensus transmission tree. We propose a polynomial time algorithm for doing this. The distance metric that we use to construct the consensus transmission tree is such that it accounts for both the topology of the transmission trees as well as the number of strains that are being transmitted. So we use the symmetric difference of the multisets given by the H sets of the transmission trees. So given these 2 transmission trees S1 and S2, at the intersection we have one edge that goes from the red host to the blue host. On the left are the edges that belong to S1, not included in the edge set of S2. And on the right we have the edges of S2 that are not included in S1. So the distance between 2 trees is given by the sum of all these edges, which is 6 in this case. So we apply our method both on simulated dataset and an HIV data collected from an HIV outbreak with a known transmission history. The simulations were run with an SIR model for the transmissions and a coalescence model with a constant population size for the within host diversity. And we run these simulations for different number of strains being transmitted during an infection and different population sizes of the outbreak. So here I show the results from the simulation data. I'm showing the size of the solution set for 3 different kind of problems. So the orange boxes for our problem, the titers problem, where we have a contact map and we are looking for transmission trees that are spanning trees of the contact map. Now, on top of this, if you add a constraint of strong transmission bottleneck, then you get what Stratus solves for. And the first observation we make is that when the simulation has a weak transmission bottleneck, Stratus fails to give any solutions for the problem instances, which means that a weak transmission bottleneck needs to be considered for inferring and sampling the solutions. Um, Further, if we relax the unique infector hypothesis from our problem, we get another set of another solution set, which is the naive solution set. It turns out that you can solve that problem and sample from the problem in polynomial time. And so this gives us an opportunity for a rejection-based sampling approach, where we sample from the outer solution set and we reject any sample that doesn't satisfy all the constraints of our solution set. But it turns out that doing this is infeasible for large outbreaks. The sampling efficiency drops exponentially as we increase the population size and the bottleneck size, that is the number of strains being transmitted during an infection. And our method, on the other hand, is able to uniformly sample the solution space for all sizes of outbreaks and bottleneck sizes. We plot here the sampling uniformity, which is a ratio, which if it is close to 1 means that we are uniformly sampling the solution space. Now, in general, it might not be ideal to summarize the entire solution space, and so we propose 2 selection criteria which can be used to prioritize the candidate solutions that are sampled from the solution space. The 2 criteria that we propose are number of transmitted strains in the outbreak and number of unsampled lineages in the outbreak. We find that both these criteria are negatively correlated with the infection recall, which is how many transmission pairs were correctly recalled from the ground truth. And since they are negatively correlated, it means that if you prioritize your the candidate solutions that have fewer transmitted strains and fewer unsampled lineages, that would increase your infection recall of your consensus transmission tree. And that is what we observe. And we get best performance for a percentile threshold of 0.01. So this is the threshold we use for analyzing the HIV outbreak. So for the HIV outbreak, we have multiple samples collected from 11 patients, all the way from 1988 to 2006. These are the 11 patients with their corresponding entry and removal time for the outbreak. And on the left, we have the phylogeny constructed for the outbreak using BEAST. Now, our goal is to, given this phylogeny and this epidemiological data, we want to infer the transmission tree over these 11 patients. So we use our method titers to generate 100,000 samples from the solution space. On the left plot here, I'm showing a heat map of the infection recall for the 100,000 samples. And and on the y-axis, we have the number of unsampled lineages. On the x-axis, we have the number of transmissions or number of transmitted strains. And as expected, they are both negatively correlated with the infection recall. And so you get high recall solutions towards the left top corner and low recall solutions towards the right bottom corner. And taking a threshold of 0.01, we get all the solutions in this black box, which when we summarize using our consensus-based approach, we are able to recover 9 out of 10 transmissions in the outbreak. And we also show that our method is robust for the choice of this percentile threshold. So to conclude, we introduce the direct transmission inference problem for estimating the transmission history under a weak transmission bottleneck for a given time phylogeny and epidemiological data. For sampling and counting of transmission trees, we establish the hardness and show a broader application of SAT. We also present a novel method of summarizing the solution space of transmission trees. In future, we plan to extend the current framework to co-estimation of time phylogeny and transmission network using a maximum likelihood-based approach. With that, I would like to thank ISCB Group for all the help and discussion during this work. This work is partially funded by these two NSF grants. I would also like to bring to your attention a related talk in the COVID-19 track of this conference on characterization of SARS-CoV-2 viral diversity within and between hosts, which will be day after tomorrow, and I invite you all to attend that as well. With that, I'll be happy to take any questions. Thank you.